A jellyfish 4.5 millimeters across, smaller than your smallest fingernail, is dying in a laboratory container. Its bell collapses. Its tentacles retract. The cellular countdown that ends every animal on Earth begins its sequence. Then the sequence stops and reverses. The body reabsorbs itself. Cells that had fully committed to adult function begin shedding that identity. What was a mature organism contracts into something earlier, not healed, not regenerated, developmentally younger. A fully formed adult returned to a juvenile state, intact and functional, ready to start again. No other confirmed animal on Earth does this. The observation was first made in the late 1980s and formally published in 1996. For nearly 3 decades after that, the mechanism sat unexplained. The event was real. The cellular behavior was confirmed, but what inside the genome enabled it remained entirely unknown, and every framework researchers proposed failed to locate the machinery responsible. Then a team of geneticists sequenced the complete genome of the organism and placed it directly beside its closest relative, which cannot do what it does. What they published in the Proceedings of the National Academy of Sciences in 2022 did not simply close the question, it revealed that the biological tools enabling this reversal were not unique inventions. Versions of them already exist in the human genome, they are just far less capable. The animal is Turritopsis dohrnii, commonly called the Immortal Jellyfish, though that name requires a qualifier. It can still be eaten by a fish. It can still be killed by disease, pollution, or temperature shock. What it cannot be killed by is aging itself. Native to the Mediterranean Sea, its translucent bell houses a bright red stomach visible at its center. It measures roughly 4.5 millimeters across. Through ballast water discharged from cargo ships over the past century, it has established populations in the Atlantic, Pacific, Caribbean, and Indo-Pacific. It is likely in every major ocean basin now. Most people have never seen one and would not recognize it if they did. The observation that placed this animal at the center of aging biology happened by accident, in a jar. In the late 1980s, 2 graduate researchers named Christian Sommer and Giorgio Bavestrello were collecting hydrozoan specimens along the Italian coast. Sommer was working through Ruhr University Bochum. Bavestrello was at the University of Genoa. They placed collected medusae into laboratory containers, expecting the standard jellyfish outcome. The adult reproduces and dies. Instead, the animals that should have been dying sank to the bottom of their containers, reabsorbed their own bodies into dense, undifferentiated masses of tissue, and over 24 to 72 hours reorganized into juvenile polyps. What entered the container as an adult organism left it as a new one at an earlier stage of development. Stefano Piraino and colleagues published the formal analysis in 1996 in the journal Biological Bulletin. They named the process transdifferentiation, fully mature, specialized cells changing what they were rather than simply dying. The committed adult cell identities that every other known animal maintains permanently were dissolving and rewriting themselves. Piraino described it as counter-evidence against the fundamental dogma of biology that lives go in one direction. That dogma had held for over a century. It was now wrong, at least for one animal, but confirmation of the event was not the same as explaining it. The mechanism behind it remained invisible for decades more. The explanatory frameworks offered to fill that gap were incomplete by necessity. Some researchers pointed to the animal's simple body plan, with no centralized brain and no fixed germline separating reproductive and body cells, arguing those structural simplifications might make large-scale cellular reprogramming energetically feasible in ways impossible for complex animals. That argument identified a context. It did not identify a mechanism. Others pointed to the evolutionary distance between cyanidarians and vertebrates, suggesting different biological rules might apply across that divide. Same problem. Nothing located the actual machinery, and the one observation that turned out to matter most was being largely overlooked. Most species within the Turritopsis genus cannot reverse their life cycle after reaching sexual maturity. They reproduce and die like every other animal. Only Turritopsis dohrnii, among the confirmed species studied, performs the full reversal. That meant the capacity was species-specific, and if it was species-specific, it had to be encoded in the genome itself, in the genetic differences between the animal that reverses and the ones that do not. Finding those differences required sequencing both genomes side by side. That work arrived in 2022 from a team led by Maria Pascual Torner, Carlos Lopez Otin, and Victor Quesada at the University of Oviedo in Spain. They assembled the complete genome of Turritopsis dohrnii and placed it directly beside the complete genome of Turritopsis rubra, a closely related species confirmed incapable of full post-reproductive rejuvenation. Same genus, broadly similar body plan, different outcome at the moment that defines the species. Every genetic difference between the two was a candidate explanation. The team also tracked gene expression across every stage of the actual reversal process. What the comparison produced was not a single mutation. It was a coordinated cluster of genomic differences spanning 5 distinct biological systems, all pointing toward the same function. The first finding was the most numerically striking. Turritopsis dohrnii carries approximately twice as many genes associated with DNA repair and damage protection as Turritopsis rubra. Specific DNA repair gene families, including duplications of genes in the mismatch repair and double-strand break repair pathways, are expanded in the immortal species and absent or reduced in the mortal one. Every time the animal undergoes reversal, cells divide and reprogram at scale across the entire organism. That process generates replication errors. In most animals, accumulated replication damage leads to breakdown and senescence. The expanded repair machinery appears to be what allows the reversal to complete without that collapse. The genome has the maintenance capacity the process demands. Its mortal relative does not. The second category was telomere maintenance. Telomeres are the protective sequences capping chromosome ends, and their shortening with each cell division is one of the primary molecular mechanisms of aging in animals. In humans, degraded telomeres lead to cells that cannot divide accurately, eventually ceasing to function altogether. That countdown is treated in most biology as a biological constant, irreversible once it begins. In Turritopsis dohrnii, the study identified structural variants in the POT1 complex, a set of proteins regulating how telomerase, the enzyme that extends telomere length, is allowed to act on chromosome ends. The specific variant found appears to reduce the inhibitory effect on telomerase. The practical result is an organism better equipped to maintain or restore the chromosome caps that most animals simply watch erode. The third category involved the redox environment. Every cell generates chemically reactive molecules as a byproduct of normal metabolism. Those molecules accumulate over time, damaging proteins, lipids, and DNA in a process directly tied to cellular aging. Turritopsis dohrnii carries significantly expanded gene sets for managing this oxidative load compared to Turritopsis rubra. During the reversal, when cellular reprogramming is generating metabolic activity at unusual scale, the enhanced antioxidant machinery appears to limit the oxidative damage that would otherwise accumulate. The animal is protected from one of the most fundamental drivers of cellular aging at the precise moment it is most biologically active. The fourth finding was the one that directly explained how transdifferentiation was possible at all. In 2012, Shinya Yamanaka received the Nobel Prize in Physiology or Medicine for demonstrating that adult mammalian cells could be returned to a pluripotent state capable of becoming any cell type by introducing specific proteins into them. Those proteins, functionally resembling OCT4, SOX2, NanoG, and MIC, rewrite the gene expression program of a mature cell to resemble that of an early developmental one. Yamanaka had to engineer that process artificially in a laboratory. The 2022 study found that during Turritopsis, Dohrnii's life cycle reversal, the target genes of those same pluripotency factors are activated in a coordinated sequence during the cyst stage. The jellyfish does not borrow this capability, it evolved it. The reprogramming machinery exists natively in the genome, requires no external introduction, and activates under physiological stress. Nobody put it there. The fifth finding concerned the polycomb repressive complex 2, and it revealed something the researchers noted had never before been documented in jellyfish. In embryonic stem cells, this protein complex maintains pluripotency by silencing the specific developmental genes that would otherwise commit cells to a particular adult identity. The complex places chemical marks on DNA that suppress differentiation, holding cells in an open, flexible state capable of becoming any tissue type. During Turritopsis, Dohrnii's reversal, the transcriptome analysis showed that the exact same pattern of gene silencing observed in embryonic stem cells is reproduced during the cyst stage. The developmental genes that would lock cells into an adult committed state are being suppressed in a configuration that mirrors early life. The molecular strategy embryonic stem cells use to remain unspecialized is the same strategy this animal uses when it decides to become young again, first documented in jellyfish here. Taken together, these 5 categories describe not a lucky accident of biology, but a coordinated genomic architecture built around a specific function. The DNA repair capacity is scaled for repeated reprogramming. The telomere maintenance prevents the chromosome countdown from reaching 0 across cycles. The redox environment limits oxidative damage during the process. The pluripotency machinery is present and activatable on demand. The epigenetic pattern for maintaining an unspecialized cell state is deployed when the reversal begins. Every relevant system points in the same direction. The genome of Turritopsis dohrnii was shaped over evolutionary time to make this possible. The trigger for that reversal has been studied more closely by one researcher than by anyone else alive. Shin Kubota, a biologist at Kyoto University's Seto Marine Biological Laboratory in Shirahama, Japan, has maintained living cultures of Turritopsis dohrnii for decades. He has documented individual specimens cycling through the complete rejuvenation process roughly 10 times within a 2-year span. He calls it the most miraculous species in the entire animal kingdom. What his observations consistently show is that the trigger is physiological stress, starvation, physical damage, temperature disruption, or age-related cellular deterioration. The animal does not reverse on a schedule. It reverses when the cellular environment signals that reversal is the better option. And the genomic architecture to execute it is always present, always ready, waiting for the conditions that switch it on. The implications for aging research were acknowledged directly in the 2022 paper. The gene families identified in Turritopsis dohrnii are not foreign to vertebrate biology. The POT1 telomere complex exists in humans. The polycomb epigenetic machinery exists in humans. The DNA repair pathways share common ancestors with vertebrate repair systems. The pluripotency-associated transcription factors have direct counterparts in human cells. What Turritopsis dohrnii carries is not an alien invention. It is an expanded, modified version of tools already present in the common ancestor of cnidarians and vertebrates, hundreds of millions of years before either existed in current form. The jellyfish did not build something new. It kept something and refined it while the vertebrate lineage let the same tools diminish. The researchers were careful to say what this does not mean. The cellular context of a 4.5 mm hydrozoan with no centralized nervous system is profoundly different from vertebrate biology. Replicating that function in a mammalian system is not a matter of identifying the genes and moving them. It is a matter of understanding a complete biological system well enough to rebuild its behavior in an entirely different organism. That work is ongoing, and the gap between the 2022 findings and practical application in human aging remains significant. What the study gave aging research was something it had not previously possessed: a complete molecular map of an organism that has solved the problem, annotated gene by gene, against a mortal relative used as a control. Every system separating the animal that reverses from the animal that cannot is now identified and documented. That map did not exist before 2022. It exists now. And what it shows is that the mechanism behind biological immortality was never hidden inside some exotic creature with no connection to us. It was encoded in the genome of an animal the size of a grain of rice, drifting in coastal water, doing something no other confirmed animal on Earth does, built from molecular tools that, in a less capable form, are already inside every human alive.